Thank you very much for the very kind introduction. One correction to make, GE heard that I'm coming to visit you, and they asked me to give the talk, so they didn't actually fly me here or brought me here.
I sponsored you lunch then.
But I do have to disclose that our research PET+MRI program is funded by GE Healthcare, so that's my disclosures. I don't have any direct financial disclosures to disclose, no pun intended, but our research program is largely funded by GE. So before I start showing you the slides that I prepared for this talk, just like Matthias mentioned that his slides were a reflection of his experience with one vendor. By no means this talk is meant to be sort of a universal presentation on how to set up a PET+MRI program. We decided in 2011 or 2012 to launch a PET+MRI program at Stanford. We traveled extensively at sites that were already using PET+MRI at that time, and I want to acknowledge the kindness of everyone in Munich, as well as at other institutions in Germany and in Asia who shared with us not only their success, but also the challenges of setting up PET and MR. And you know, I picked up this title, obviously, as a reference to how to get away with murder, because at times it did feel like just like that in setting up a PET+MRI program. And you also, I hope you noticed that I didn't put PET-MR, I didn't put Mr. PET, MR PET, I didn't put / between them. I put plus, because I really think that if you are to have a successful PET+MR program, it has to be a collaboration between PET experts and MR experts. There are very, very few people in the world who are experts in both. So in any program, you have to make it such that people on both sides feel like it's a powerful collaboration. It's something that both parties get something out of it. And ultimately, that is the only way to result in a successful program. And the last thing I'm going to say before starting the slides, a good friend of mine, Roland Scholl, who's Australian and used to work for a competitor of GE, said a joke that to this day I really say it whenever I have an opportunity because I really think it's good. He told me that when they installed the first MMR in Seoul, they moved the DMZ between nuclear medicine and radiology. So we don't want to have a DMZ between radiology and nuclear medicine. We want to work together because it's the only way to advance the field. So a brief introduction. Again, this is about the research PET+MRI program. It's not about the clinical PET+MRI program that we have. And then I'll finish with a few developments related to what has been done in PET+MRI to date. Just to put in perspective, the Stanford healthcare system, it's in the midst of a significant growth, and that's because of the healthcare system in the US that requires you to have a large captive population of young, healthy adults that pay insurance, so ultimately you can provide high level of care to the elderly who have high needs. So because of that, the hospital expanded significantly with new buildings, new cancer centers. In fact, the two buildings at the bottom, the new children's hospital that will open next year and the new adult hospital that will open in 2018 are the largest healthcare project in the US at a total cost of about $6 billion. And the neuroscience here, it's a new building that integrates everything that has to do with neuro, from imaging to stroke to cancer, in a single building, with the idea that the patient will come see a specialist and they will go have their imaging done, return, and everything will be done. That's where one of the PET+MRI for clinical use is actually installed. In nuclear medicine, really everything started to take shape as a competitive arena when Sam moved from UCLA, and at that time, the Lucas expansion that's housing the cyclotron was built, as well as the Clark Center, which was a wonderful idea because people in the dean's office noted that scientists in engineering, in chemistry, in medicine were working on the same projects, but without knowing about this. So they would not talk to each other. So this building has all glass walls to signify transparency and forces people to work together from various areas in the university. So it forces people from engineering, from chemistry, from medicine to be sharing lunch in the same cafeteria or sharing the small imaging facility, and it's really the only way to increase turnout. And lastly, our clinic opened here in 2010 in an ex-surgical space. And it was designed so that if you close a set of double doors, you can access it from the back and do large animal experiments, for example, without interfering with the clinical activities that are done in the front of the clinic. Our initial idea was to place the PET+MR right here. We don't have windows like you have at Peter Mac, but we're on the second floor, so we're not in the basement. We're somewhere in between, not as good as you are with windows, but not in the basement. One challenge was the weight of the PET+MR system, and turns out that the weight was okay to be supported on the second floor with a little bit of reinforcement, but the other issue was vibrations. So it turns out that the way the hospital is built, vibrations from this room would go all the way half a mile on the other side of the hospital. So clearly that was not a feasible enterprise. So we installed a research PET+MR in another building that's purely research, which also shortened the approval time by quite a bit. And why PET+MR makes sense at our institution? We have two large programs, the molecular imaging program at Stanford MIPS that's led by Sam, and the radiological sciences laboratory. So anyone who knows MR probably knows Gary Glover, who's really one of the experts in the area. And both these research PET and research MR programs are very successful. So bringing them together in a PET+MR program for research activities made a lot of sense at that time. So what is available? How do you make your choice? Well, a lot of things go into what vendor you pick. I think the most important should be what technology is available there, but I recognize that other factors, such as your prior relationships, be it commercial or research, how your service is available, a lot of things play into it. At the time when we made our decision, the scanners available on the market were the Philips Ingenuity PET+MR, which we decided against because it wasn't a true simultaneous PET+MR. The images are great, but they're not simultaneous. And of course, Siemens Biograph MMR was available at that time, resulting in great images, as the group in Munich published as an early experience with it, comparing with PET-CT. However, we felt that technologically there's more coming, and we were willing to take the risk of waiting and having phone calls with our partners, pushing them to move faster. Because this silicon photomultiplier technology was showing very promising results. As I said earlier, the MMR is indeed simultaneous, but it doesn't have time-of-flight ability, and you would wonder why is time-of-flight important. We don't use time-of-flight routinely in our clinic. We only use it for large patients. Data from Joel Karp and the others at Penn indicates that really time-of-flight with the timing resolution available in PET-CT at that time was only meaningful for large patients. But we felt that with this new technology, the timing resolution is also going to be better and is going to have an impact on image quality. This is a short video demonstrating how these SIPM detectors work. So you have a base where these are installed. This is the crystal coupling them. Of course, you need to shield them from the magnetic field. You install them in PET+MR in a 25 centimeter axial field of view. You have cold water, keeping them at a constant temperature. You have fiber optics on the other side, integrated with a body coil, and ultimately in a product that looks just like an MR, but it is a PET+MR. One very important aspect of this system, and this is data from the technical brochure from GE, is the high sensitivity, 21 counts per second per kilobecquerel, which is truly remarkable. It's a combination of the 25 centimeter axial field of view, but also the very sensitive SIPMs. Trust no one is our MO at Stanford, so we repeated the testing in-house, and in fact, you see the sensitivity is higher what we measured with EMR on or off versus what's in the brochure. And I always joke with my friends at GE that they're like the airline industry. You know that it doesn't take 6 hours to fly from San Francisco to New York. It's 4.5, 5 hours, but they put 6 hours, so you feel so good when you arrive early. So similarly here, you have a sensitivity that I think on purpose was listed a little bit lower than it actually is. So, when your partners are measuring it in-house, you see it higher, and you're like, wow, even better than what's in the brochure. Another good thing about this is if you have a 3T750W, so a wide bore magnet, you can remove all the plastics, the existing body coil in it, This is actually what we bought. We had at 750W at the Lucas Center, so we bought the PET ring. And in a course of a few days, and as Murphy's Law has it, this was supposed to arrive at Stanford on a Monday, early afternoon, Monday morning. The utility crew started digging a trench in front of the delivery door. So this had to be transported from a big truck on a small truck, and they started working on Tuesday. But very encouragingly, by Friday afternoon, this was installed, tested, and ready to go. This was in December 2013. And this is how it looks now in our PET+MRI research suite. Of course, the first thing, the boring thing, you have to show that this is at least equivalent to PET-CT. So we did a comparison, and unfortunately, the ethics committee did not allow us to do PET-CT followed by PET+MRI in a subgroup of patients, and vice versa in the other. So every patient had PET-CT first, followed by PET+MRI. PET-CT, as you see here, was done about 70 minutes after injection, and PET+MRI followed after about 3 hours later. And the duration of PET+MRI for this oncological patient was about 50 minutes. There was no lesions that were missed in PET+MRI compared to PET-CT. But as you can imagine, some of these patients were elderly, sick patients, so there were occasional motion artifacts on the MR component of PET+MRI. But even those were only 7 of the 52 cases. SUV values were higher, partly because we waited longer. We know from Professor Alavi's work at Penn that dual time imaging uptake increases in cancer, decreases in non-cancer lesions. But partly, this increased uptake, I believe, is related to the new technology used in this particular PET+MRI. And this is proven, I hope, in these graphs where we normalize the uptake to background that shouldn't change too much over time, such as cerebellum or liver. There's no significant findings in this study, but it's the first ever patient scan on a SIGNAP PET+MRI outside of the factory. We were very excited to notice the image quality being quite good, in my opinion, and this compared to the 690, so what was the state-of-the-art PET-CT at that time. And this is almost 3 hours after injection. I don't know how it projects. I think it projects quite well. Actually, there's nothing in the lung here on PET. You can probably recognize the mediastinal lymph node. However, when you look at the images from PET+MRI, there's clearly something that the arrow is pointing at, as well as a lymph node. So image quality in our evaluation of this study was clearly superior in the PET in PET+MRI compared to PET in PET-CT. And this is another example illustrating the same thing. You see a few nodules on the image on the left acquired on the standard BGO Discovery 600, but clearly you see much more. In addition to that, and that's something that continues to impress us and open new avenues for the use of the scanner, it's you can follow actually the spinal cord all the way to the lumbar area. And the movie is not working, but if you look at it in sagittal view, you can follow the spinal cord all the way to the lumbar area, as I said. Time of flight actually is quite important for this PET in PET+MRI because it's below 400 picoseconds. And at this timing resolution, you start seeing improvements that are not seen with a time of flight at 600 to 700 picoseconds as is standard PET-CT. And this is a great example from one of my colleagues where the attenuation correction, which is of course one of the issues that's perceived for PET+MRI, identified the error in the rectum as something else and overcorrected. Once you apply the time of flight, that artifact is gone. Also, the time of flight makes small lesions much more conspicuous, as I hope this slide illustrates it. On PET-CT, I don't think that you can call something prospectively. However, even without time of flight, the PET in PET+MRI makes that lesion more conspicuous. If you apply time of flight, that's clearly something there and something that's visible. So now, how do we use this machine? We know it works, we know it has great performance, but how do you set it up as a research program that's successful? Because, we're not going to have research funding from GE forever. This has to live on its own in a few years. So we decided to bring together people from both the PET program as well as the MR program, clinicians working with us in various fields, and we also looked at what others are doing, what are areas of imaging that are meaningful for PET+MRI. So we set up our program with 4 components. One of them is to look at neuro applications. One of them is to look at cancer and vascular applications. Another one that's, I think, interesting and to date unique is to look at pain, and we love to refer to them as a pain team. And the last team is a technical team that has to do with refinements in the performance of the machine. I am co-leading the PET+MRI research program with Gary Gold, who is an MSK radiologist, and Praveen is our deputy director, who makes everything working. Pretty much that's his role, to make sure that on a daily basis we have everything in place to have this program functional and able to offer our research collaborators a device that functions as it should be. We have many, many collaborators from various parts of imaging, as well as from various parts of medicine at Stanford. And we also established a cost center. So in addition to the sponsor research that is being done, anyone who wants to do PET+MRI research at Stanford can go to our website, input what the protocol is. They can clearly see and transparently see what is the cost of using the machine, what is the availability. They can request scanner time, they can request radiopharmaceuticals. So going forward, that and grants from NIH and other entities is what's going to keep this research program going forward. The first project that we started, as I mentioned, we called it the PET+MRI Advanced Research and Development part. And as I said, they're neuro, body, vascular, pain, and technical. But there are also other projects looking at tracer development, such as tracer development for Parkinson's disease, hyperpolarized MRS, and that's one area where I really think that having simultaneous PET and MR is very important, as well as PET and MR for myocardial tissue characterization. These are some of the projects included in this early program, as well as who are the leaders using it. And I'm going to try to show you a few examples of the work that has been done to date in this program. We also believe, and I hope that you got that message when I said that I believe this is PET plus MR, not PET/MR. We believe in fostering collaborations. Not a single institution can have access to all the patients that are needed to advance the field. Not a single country can do that. So we try to foster collaborations, and it's a small place 20 miles north of Stanford called UCSF. You probably heard of it. Anyway, it's a great institution, and we have many friends and colleagues there. But what we do right now, we have bi-yearly joint conferences with them for the PET+MR program. The first one was held in China Basin, so where the PET+MR is installed at UCSF. The second one was done at Stanford. And we exchange ideas. And you would be, why would you want to do that? Well, it turns out that it's very useful because we tackled almost exactly the same issues, but coming to these issues from very different angles. So it's good to see how others are thinking about solving issues in PET+MR, and it's validation that what you work on is actually what may be significant. It's also very important for us that we outdrink our colleagues at UCSF. So this is a group from Stanford at the first meeting, and everyone from UCSF was home already. We were still at the bar there. But as I said, it's not just regional, it's also international collaborations. We are setting up together with UCSF a CME course, and we also collaborated, and we were fortunate in that Gary, my colleague in charge of the PET+MR program in MSK radiology, he was the president-elect at ISMRM, and Hossein Jadbar, who was the president at SMMI last year, has very good connections with Stanford. He was a resident at Stanford. So we were able to get SMMI and ISMRM to work together, and we already had two very successful seminars on PET+MR, one in Baltimore last year and one in Singapore this year. So the idea is you alternate these joint symposia between 1 year at SNMI, the next year at ISMRM, because we don't want the mistakes from the PETCT infancy to be repeated. We want to work together and make it a collaboration. We need data for the PET+MR to be accepted. If everyone is doing small little protocols that have nothing in common with what others are doing, we're not going to have sufficient data to actually make this a clinically accepted tool. So we share all our protocols with our colleagues and collaborators who want to use it, and we're willing to share this data with whoever is interested in it. One of the first projects, and again, we did this because we didn't have access to PSMA or Bombazine at that time, or choline or acetate. We wanted to see, can we accurately stage patients with advanced breast and prostate cancer using just cheap sodium fluoride and FDG. To date, we actually have close to 100 patients who are referred to have a bone scan, and we intercepted them in our clinic and asked them, are you willing to have a PET+MR? What's really important here, look at the doses that we administer to these patients. So sodium fluoride, only 1 millicurie. FDG, only 4 millicurie. I mean, these are significant reductions in the administered dosages, and that's because, to one of the prior points, PET actually is acquired continuously while the MR data is acquired, so we have 4 minutes per bed, dictated by the MR sequences, but also because this machine is very, very sensitive. How the images look like? They look exceptionally good, in my opinion. Again, this is a 1.1 millicurie of sodium fluoride, 4.3 of FDG in what we like to call a cocktail study, and we know we're not alcoholics, but we saw that that's a catchy word, combined sodium fluoride and FDG cocktail. And again, 1 millicurie here, 4 over there. And we are able, obviously, it's perhaps an unfair comparison against all planar bone scan, but that's what's reimbursed and approved in the US, and that's what's used by the clinician. So we have to make our comparison to what's standard of care right now. Perfect image co-registration in this particular case. We do the coronal T2-weighted imaging. We do use the PET+MRI in initial staging of prostate cancer. I think that for biochemical recurrence, the PET+MRI was indicated here because really this was the best PET that was available at that time. But in initial diagnosis of prostate cancer, you do need both PET and MR for accurate anatomic delineation. And our study again requires that this patient have intermediate and high risk and negative conventional imaging. This is just one such examples where I think that you can clearly see that there's presence of uptake in the prostate gland, as well as in additional lymph nodes. You've seen earlier the data from Bombazine PET+MRI, so I'm not going to spend too much time. This is indeed the image that Professor Higgs mentioned earlier. I wasn't necessarily going with this image to show that any tracer is better than the other. As I was going after the fact that in the US, patients have to wait a very, very long time to have access to these tracers. I mean, this is over 40 months that this patient continuously had biopsies based on MR that were false positives and anxiety, and the patient was willing to go to Munich actually to have the PSMA when we said, hey, we can do it here now. So I really think that these agents are game changers, both Bombazine and PSMA in the United States. Another area that's very interesting to me, I know that angiogenesis is not as fashionable as it was a decade or so ago, but we do have an angiogenesis agent, an RGD peptide, a dimer, and we think that it can differentiate responders from non-responders as early as 1 week. This is the primary lesion here. No significant change at 1 week, and this clearly has progressed at the standard of care 6 weeks post starting Avastin in these patients with recurrent glioblastoma multiforme. However, let's look at this other example. Clearly there's uptake, there's angiogenesis going on in the left frontal lobe. As early as 1 week, signal is gone and is no longer seen at 6 weeks. Turns out this patient is still alive at 34 months after starting Avastin. So it's a small pilot phase study, but not necessarily the change in the actual uptake as the change in the angiogenesis volume seems to be predictive of outcomes in this patient. And of course, Avastin and other anti-angiogenesis drugs can be used in other cancers like colorectal, renal, cervical, ovarian. So I think that this has potential. This graph just illustrates what I said earlier. It's only the uptake in the lesion that significantly decreased at 1 week. The signal in other areas is not significantly changed. We also wanted to look at cardiac sarcoid, and patients who were referred for PET-CT were also imaged with PET+MRI using FDG. MR is great because it allows you to do such measurements as quantitative left ventricle estimation, right ventricle. And it was very interesting. I have yet to have a chance to speak with Professor Alavi about this because his teaching is if the uptake is going up, it's cancer. If it's inflammation, you shouldn't have higher uptake on delayed imaging. But look at this example here on the top. You have uptake in the myocardium that was not seen on the earlier PET from PET-CT. And this was the case about half of the patients in our cohort. And this was, in this particular case, correlated with enhancement on MR as well. It's not the only example. There's absolutely no uptake on PET from PET-CT, but there is more focal uptake on PET in PET-MR, which was done a little bit later. You probably have seen the publication from a group in Australia looking at Neuraseq for cardiac amyloid. It came out in the most recent issue of JNM. They looked at CHF of other causes versus CHF because of amyloid deposition, and they clearly shown that using the amyloid agent approved for Alzheimer's disease, you can actually differentiate between the two. We took a different approach here. We do a whole body scan, a whole body PET+MRI, not just the heart, and because some of these patients have systemic deposition of amyloid plaques, and that's critical for the clinicians to know. This is the biodistribution study for the Neuraseq, where you don't see uptake in areas that I'm going to show you in this example. This patient with suspected amyloid had an FDG scan, and these are images dedicated cardiac in one single bed after injection of Neuraseq, and this is a whole body exam. And if you look at the biodistribution of amyloid agents, they shouldn't really be seen in the bone marrow, and this had to do with the light chain deposition in the presence in the bone marrow. What's interesting, and that was biopsy proven in this case, what's interesting is, as I said, it's not just the uptake in the myocardium, but in this case, there's also uptake in the stomach wall. And you can say that what you see in the bowel, it's probably hepatobiliary clearance, but it would be a little bit difficult for that to reflux back into the stomach. So we did not believe that it was that. The patient had an upper endoscopy, and this was biopsy proven to be amyloid deposition. So new avenues that are opened up for traces that are already approved and can be used off-label, and a lot of what we do in nuclear medicine is off-label use of FDA-approved agents. This is a neuro study from the neuro team looking at C11 raclopride, and I think it's one of the most beautiful images of the brain using PET, the definition of these structures. It's absolutely incredible, but that's not the only thing that's interesting. My colleagues, led by Gary Glover, are looking into doing functional PET, so they do a reward punishment test with this patient, and of course with MRI, that's what's functional MRI, you can see displacement of the signal, but for the first time, they were able to show displacement of the signal on PET, so displacement of the raclopride by dopamine release in response to the stimulation with the patient done in the scan. So you can only do that because you have very high temporal resolution and sensitivity of this scanner. I mentioned MSK applications, so I'm going to try and play this movie and take a look at the PET image over the lumbar area. So in this bed here. That image is acquired over 40 minutes because that's how long it takes to do the MR in that area. It's the first time and the only time that I was able to actually identify nerve roots on FDG PET in this patient. Again, I think that speaks for the high quality of the PET in this machine. We asked our radiologist and PET physician to tell where's the pain coming from, based on MR or from PET-MR. And if you look at MR only, it's actually worse than flipping a coin. They were correct in 5 of 14 cases. But if you look at PET-MR, they were correct in 12 of the 14 cases. And not only that, they were actually able to act on that. So there were ganglion cysts that were muscle that were inflamed, nerves that were injured. So they were able to actually do injections, do surgery, etc. And so they're able to remove the pain for these patients. So who would have thought that pain can be a killer app for PET-MR? It's not proven yet, but I think that's one direction where more and more people are going now with PET-MR. And there's sigma receptor agents that have been published now trying to identify pain. And I think that that's something that will be very good for PET-MR overall worldwide. Now, as far as latest developments, I wanted to show a few slides. We also just installed in August the first SIPM-based PET-CT. So the same detector as in PET-MR, but now translated to PET-CT. In our particular installation with only 4 blocks, so 20 centimeters axial field of view. This has a 70 centimeter bore, so it would have been much more expensive to do a 25 centimeter extension of these detectors. We just had our ribbon cutting ceremony relatively recently. This is some of the calculations, so pretty high sensitivity according to our internal NEMA testing. The timing resolution, really good, 374 picoseconds, so as good as in PET-MR. As it happened, the first patient was scanned when I was at WMIC in New York, so one of my colleagues was teleconferencing from the console showing me the image, and so that I was able to see the first PET done on a PET-CT as it was acquired. And not much special about here, but this is the first ever image acquired with the digital, with this SIPM PET-CT, the Discovery MI. And many times you expect when the first thing is done to need to apply filters or change some of the settings, but this image is actually pretty darn good, and it was without any twisting of the device, without anything else. Since then, we've conducted a pilot phase study evaluating the quality of PET images from the standard PET-CT versus SIPM PET-CT. So this is a standard reconstruction from a time-of-flight Discovery 690, standard recon from the Discovery MI, with time-of-flight and now with Q-clear, so with multiple iterative reconstruction. And I think that here you start seeing a lesion much more conspicuous and definitely conspicuous in the internal mammary chain, definitely better seen when you apply Q-clear. But take a look at these patients with melanoma. There's nothing in the liver that I can call on the image from the 690. You start seeing something, perhaps even with a standard recon. Once you apply the time of flight and the Q-clear, there's definitely something in the liver. Now, of course, the issue is what if that's inflammation? What if there's something that's meaningless? What if you start chasing our tails now for things that are insignificant for patient care? In this case, it was significant because it was a metastasis. Another thing that we notice is that the improved timing resolution, and this is good both for PET in PET-MR and PET in PET-CT, will result in a help with some of these respiratory motion artifacts. You can see the artifacts under the diaphragm. This is without applying any gating. This is simply because the timing resolution is better in these SIPM-based detectors. You tend to eliminate these artifacts. And this is what really got us very excited. This is a patient with lung cancer treated with surgical resection, and not much going on the standard recon. The standard recon, clearly you can see there's a right hilar lymph node, but we reconstructed the image at 2 minutes per bed, that's 30 seconds per bed. So look at the 30-second per bed image. You don't miss any of the diagnostic quality from the other one. So who's going to read all these scans? If we can do 10 PET-CTs in 1 hour, how many people do we need to hire to read the scans? So I think that it will be a combination of significantly decreasing the dose of administered radiopharmaceuticals and also increasing throughput. So we're already thinking that perhaps in the room that houses the BGO scanner, we can install a few more chairs for uptake time, because the amount of patient being scanned is not going to be an issue. I'm going to skip a little bit quicker here. We asked 2 of my colleagues to look at the scores for images from Discovery MI with time of flight and Q-clear versus just time of flight versus non-time of flight versus standard. And they were blinded as much as you can be blinded, because after a while you can recognize clearly what image is coming from where. The scores were definitely higher when you apply both time of flight and Q-clear. Another thing that's really exciting is gating. How many people in the room have gating devices on their scanners? How many people are using them routinely? Okay. So not everyone. We have it. We don't use it because it's cumbersome. It requires another image acquisition. Here with device-less gating, this image on the right, it's corrected. The PET data is corrected simply from what's in the image, and this will be available at some point, both in PET-CT and PET-MR. And you can clearly identify the patient with sarcoid. So those are sarcoidosis lesions in both the liver and the spleen, and the image definition is definitely better. Just briefly about Q-clear, what you need to know is that you can do 25 iterations while you suppress the noise, and that will bring the signal closer to what it's supposed to be. Both of these readers gave higher scores, again, blinded to the Q-clear reconstructions versus the standard reconstruction. But more importantly, if you look at the Q-clear reconstruction, you can start seeing like this femoral lesion that was biopsy proven to be metastatic disease that was simply not identified on the standard recon. This was actually a scary example. This GBM patient, where we clearly can identify the recurrent GBM on all reconstructions. But that dot, what is that? You only see it with Q-clear. You can't really even imagine it on the standard recon. Is it just noise, or is it something real? Unfortunately, for the patient on the MR done 1 month later, that was something real that showed up on MR at that point, and it continued to grow at the 2 months follow-up MR. So I really think that if you have access to things like Q-clear, you have to use it it's disruptive to your workflow because, you know, all of a sudden the values are going to be higher. What you measure, clinicians will not know what you're referring to. But at one point, you have to make the transition because otherwise, you're just going to miss out these lesions that can be significant for patients. And for dotatate, since we are the theranostic here, clearly a lymph node in the internal mammary chain, we're much more confident calling it with Q-clear than we are calling it with a standard recon. And as I said, this is available in PET+MRI as well, and you can see time of flight, PET from PET+MRI, after applying Q-clear, there's better lesion clarity as well as reduced noise. So, you know, the team has made significant improvements in image quality above what I really think that it's a great hardware to begin with. And probably you are in the room, you've seen this about the gallium. I'm very proud that our team working with the GE engineers were able to actually solve this issue of scatter correction, which is a major problem for gallium agents that tend to accumulate in higher concentrations in bladder. In this case, I believe the ratio of uptake in bladder to soft tissue was something like 600 to 1. So that's what results in these significant scatter correction artifacts, and they're gone. So I'm not going to spend too much time because you've seen them already. How many people believe this? That you do PET-CT MR at 50% discount? I don't believe it either, but it was so funny. When I visited my friends and colleagues in Zurich, across from their center, it's a supermarket, and they had this sign right under the sign for PET-CT and MR, and I felt that, you know, I will take a picture and show it to my colleagues and advertise for Zurich, the most expensive city to live in Europe, whether you believe it or not, PET-CT MR 50% discount. I want to acknowledge my colleagues who do a lot of the work in nuclear medicine at Stanford, as well as everyone in the department. It takes a village to make something durable, and definitely, if you are contemplating PET+MR, make it a collaborative effort between everyone who will use it, otherwise it's not going to work. For us, it worked. We now have 3 more machines, either installed or in the plan. One is already installed in the neuroscience building. The other one will start working next year in the new children's hospital. And the first one will be at the Veterans Administration Hospital in Palo Alto, where I think a lot of interesting work related to traumatic brain disease is going to be done. So I want to thank everyone for being here, the organizers for allowing me to give the presentation, and GE for inviting me to speak. Thank you very much.
Just down the road from here, there's Howard Florey Research Institute. Howard Florey was one of the co-discoverers of penicillin, and a book about him said his most enduring research project was a search for money. And I think Sam's obviously found the recipe to that. He seems to find the money fairly well. And I think it's a really important message that for this group who are in searching always for better traces with higher contrast and higher sensitivity for detection of disease, and that's going to be important for planning the therapies, to realise that hand in hand with that we have ever-improving technologies and better software for displaying them, and which means we're going to see smaller and smaller lumps and be able to characterise those lumps better and better. And as someone's pointed out, if you can see it, you can treat it, but the really important question is everything we see important? Do we need to treat it, or can we afford to watch it? And you know, the poker met for the baby boomers, I thought a better game might be whack-a-mole, where you hit it here and it pops up there, and you hit it there and it pops up somewhere else. But it's a very important area, I think, for us to start to get beyond lumpology and start to think about biology and using the best tools to do that, whether they're tracers, better imaging equipment, or better software. Any questions from the audience? If not, thank you very much for your attendance. Oh, sorry, Rob. So there is one question.
Just one question. Yes. In the early days, it was a little bit difficult, for instance, in the brain, to do the attenuation correction of the skull. How is it solved in the industry?
So every vendor had to struggle with this, and a neuro person, but I appreciate the neuro people's pain of having to have very accurate measurements. My philosophy is that I'd rather have the same error every time rather than random error. So if the patient is imaged every time on the same machine or a similar machine, the percentage change is what's mattering, not the actual number. I would be much more uncomfortable with measurements with different errors. To answer more precisely to your talk, right now the GE device has both the MR-AC sequences as well as an atlas-based method to correct for head and neck. And one development that's just coming to the market, it's a ZTE, so a very fast MR sequence that's actually able to create the bone mask that can be applied, because really the bone is the issue. MR is not seeing bone. So efforts are made by all vendors to advance this, and I think they're going to be successful with that. Thank you.